And I'm not much for introductions. I mean, the following two speakers are both phenomenal in their own rights. There's going to be some really interesting talks. Lluis, I met last time. I was sitting right next to him. He didn't recognize me, but I recognized him because I've been following his work for a long time. And he gives phenomenal talks. And so I don't want to take any time away from him since we're on a time schedule. Lluis, can you please come on up and talk about immune gene selection in human evolution and some really fascinating updates, and they're doing some really phenomenal work. So, thank you, Lluis.
Thank you very much for inviting me here today to give you a bit an overview of what we have been doing in the last years and how we can use also the other way around. Mara was talking about how to use microbes to understand humans, and basically what we are doing is to use humans and natural selection to understand a bit more microbes and how microbes have exerted pressure on human genome diversity. So basically my lab is interested in reconstructing a bit the demographic history of some human populations. In particular, we have been interested over the last years in Central Africa, and we are increasingly interested in understanding the last pieces of the globe that were peopled by modern humans, that is Melanesia and Polynesia. Basically, humans arrived to Polynesia only 1,000 years ago. So it's the very, very last chain of the human peopling of the world. But also, we're interested in understanding how during all these dispersals around the globe, and the admixtures that these populations have either with ancient hominins like Jean-Jacques told you this morning, Neanderthal, Denisovans, or modern admixture between modern human populations, how all these travels and migrations, populations adapted to pathogen pressures. So basically, our idea is to try to understand how pathogens have exerted pressure on the human genome, to understand immunological mechanisms that are essential, and others that are less and more dispensable. So the idea is that why do we care about natural selection and infectious diseases? As you know, infectious diseases have been a major cause of mortality across all human evolution until yesterday from an evolutionary point of view 100 years ago. And on the other hand, we know also that there is a huge amount of immune variation between us, either from the same population or from different populations. So using different approaches, like population genetic approaches, functional genomics, and systems immunology, what we are basically trying to understand is the genetic, non-genetic, and evolutionary factors that drive immune response variation in humans. So in terms of population genetics and evolutionary factors, which we have been trying to identify how different forms of selection have impacted the human genome, and how today genes that evolve under purifying selection or positive selection are going to be involved in immunological mechanisms. Basically, purifying selection means that you have genes in the genome with many mutations. Most of mutations are neutral, but some mutations are coming, that these mutations are deleterious, and these mutations are removed from the population by purifying selection. Purifying selection purges out of the population deleterious variants. Whereas adaptive evolution, like one example is positive selection, where you have lots of mutations in the past, neutral, most of them in blue, one mutation is in red, and this mutation, let's say, provides you a selective advantage against tuberculosis or malaria, so this mutation will increase in frequency in the population. So we have been showing that by dissecting the intensity and type of selection today acting on immune genes, we can predict how these genes are going to be involved in immunological mechanisms or even in diseases. Genes evolving under purifying selection are essential and non-redundant, and these genes are predicted to be involved in severe disorders. Whereas genes evolving adaptively, in generally, are genes where novelty and diversity is favored, and they fulfill functions that have participated in human adaptation. So we have been applying this to innate immunity, our first line of host defense against pathogens, and we have been dissecting the type and intensity of selection acting in around 1,500 genes. This is just a summary of many of the work we have done over the last 10 years, I would say. And we have been able to distinguish, depending on this intensity of selection, genes that are essential and non-redundant, like TLRs specialized in the sensing of nucleic acids, from genes that fulfill redundant functions, like the sensing of flagellin from flagellated bacteria. But also we have been identifying adaptive mutations, mutations that have conferred us a selective advantage, like mutations in type 3 interference that have conferred selective advantage, in this case, to Europeans and Asians, mutations in the TLR1-610 cluster associated with a reduction of the inflammatory response and specifically providing a selective advantage to Europeans, and also mutations and haplotypes that we have inherited from Neanderthals. We have, for example, shown that the TLR1-610 cluster, together with the OAS cluster involving antiviral responses and some interferon genes, are very enriched in Neanderthal ancestry, meaning that through archaic admixture with Neanderthals, we acquire advantageous phenotypes. Also, we have been shown that the bulk of human adaptation to pathogen pressures, at least with what regards to innate immunity genes, occur in between 3,000 and 13,000 years ago, which coincides with the period in which populations started to shift from being hunter-gatherers and to being sedentary farmers. But all these are signatures of selection, are molecular genetic signatures. So the next question is, can we learn a bit about the adaptive phenotypes? So for that, we have been focusing on immune responses as a potential adaptive phenotype. And basically, our general rationale of all this is that we study individuals, either from the same population or from different populations, and from these individuals we have their cells that we can either purify or we just use whole blood, and we infect these cells with either live pathogens like BCG, that you know that we use also as a vaccine for tuberculosis, influenza virus, or just ligands that are going to activate our innate immunity. And then basically we do, for example, gene expression before and after infection, and then we map the genetic determinants of gene expression responses to bacterial or viral infection. So we have been doing this in the context of a project called Evo Immunopop, in which we have 200 individuals, half African ancestry, half European ancestry, all living in Belgium to homogenize as much as we could the environmental exposure. So from these individuals, we purify their monocytes, a major innate immune cell, and the monocytes were stimulated with 3 different TLR ligands, activating 2 pathways called TLR1-2 and TLR4, mainly involved in bacterial responses, 1 pathway called TLR7-8, mainly involved in antiviral responses, but also we use a live strain of influenza virus. So we sequence the RNA to understand how genes behave before and after infection, and then we map the genetic determinants of gene expression variation, and as you will see, we apply a number of population genetic tools. So first thing that we saw is that gene expression is mainly driven by the immune condition. Here you have non-stimulated cells, cells stimulated with influenza virus, and different cells stimulated with the different ligands of the different TLR pathways. When you zoom in one of the conditions, like the TLR1-2 pathway, you can start to see that there's a slight differences in terms of immune responses between Europeans and Africans. And basically what we show is that there's obviously none on-off differences between Europeans and Africans in terms of immune responses, but what it changed markedly is the intensity to which they are going to respond to some of these immune stimulations. We also show that in most cases, these differences in intensity are due to genetic variants located in cis that are going to control gene expression in a stimuli-specific manner, meaning that in the absence of immune stimulation, the genetic variants do not have an impact on gene expression, but these variants will have an impact on gene expression only in the presence of infection or immunostimulation, highlighting gene-environment interactions. These marked differences in the intensity to which Africans and Europeans are going to respond to some stimuli sometimes can even be explained by a single mutation. We identify a number of master regulators of gene expression in interferon beta-1 or TLR1, and for example, the case of TLR1, we identify a mutation that is present in 2% of Africans, 70% of Europeans. This mutation is associated with a drastic reduction in NF-κB activity, but most importantly, this mutation is associated with the decreased expression of around 100 genes involved in inflammatory responses. So, in other words, 70% of Europeans have a mutation in TLR1 that is a master regulator that is going to be associated with a dampening, with a lowering of our inflammatory responses. And moreover, this mutation presents one of the strongest signatures of positive selection in Europeans, suggesting that reducing the inflammatory response has been a selective advantage for the host. More generally, what we also showed here is that the analysis of positive selection allows us to identify regulatory variants associated with gene expression variation in the presence of immunostimulation that have conferred different selective advantages to Africans and Europeans. And for example, we found another example, in this case in African populations, of how reducing the inflammatory response has been an advantage for the host. But also by overlapping this mapping of regulatory variants associated with gene expression variation in the context of immune responses with variants that we have inherited. By we, I mean us of European ancestry, the ones here that are of European ancestry. You know that 2-3% of our genomes comes from Neanderthal. So what we have shown is that regulatory variant controlling gene expression, in particular those associated with antiviral responses, are enriched in variants that comes from Neanderthal ancestry. But not all is about just gene expression. There's other more fine-tuned way to control immune responses following infection, and a kind of very cheap, easy, fast way, because it's basically with the same material you have in a gene, make different proteins, as you know, is alternative splicing. So we have been defining the alternative splicing landscape of monocytes, this major innate immune cell type. We have in Africans and Europeans. We have detected around 16,000 alternative splicing events, most of them being exons that are being skipped. That means that you are going to have a shorter protein. We've seen that just using the different isoforms, we are recapitulating quite well the different conditions of stimulation, non-stimulated, flu, and the different TLR pathways here. But most importantly, what we are saying is that, for example, NF-κB is differentially expressed upon infection, obvious, and there's a change in the isoform being used upon infection, why not? But most importantly, we have identified around 1,000 genes that they are not differentially expressed before and after infection at the gene unit level. So you completely miss them when you are doing a kind of basic study of transcriptome analysis that you are using your bacteria or your virus, and you are looking which are the genes that are differentially expressed. These genes are not differentially expressed, but the main isoform that is used before infection and after infection is completely shifted, right? Highlighting the interest of looking at alternative splicing. We have also mapped the genetic basis of alternative splicing. And to make a long story short, because we're getting now to genomics and less evolutionary, what we have shown is that the genetic determinants of alternative splicing are mostly independent of the genetic determinants of gene expression. But an evolutionary example of that is this gene that the isoform usage of this gene is under genetic control. When you have a CC here, you are including an extra exon in the gene, and the inclusion of extra exon is under a strong positive selection, particularly in European populations that are going to have a form of this gene with this extra exon here. Again, it's one of the strongest signatures of selection at the genome-wide level of Europeans. And when you look what is doing this C allele, basically this C allele is associated with higher protection against pediatric tuberculosis, despite the same allele is associated with increased susceptibility to allergic rhinitis. Again, showing now this trade-off, as has been shown this morning, between past selection against infection, despite the price to pay today, in some cases can be increased susceptibility to allergy, inflammation, or autoimmunity. But not all is about basic population genetics principles, like positive selection. Not all is about genetics even. So some of the questions that were posed in the last years is that, is there other forms of adaptation to pathogen pressure? What about epigenetic changes? And also what about changes in lifestyle? So we've seen that we, Europeans and Asians, admixed around 40,000, 50,000 years ago with Neanderthals and Denisovans. We've seen that we acquired from them some HLA haplotypes that have been adaptive, some TLR forms, so some interferon forms. But there's something much more simple, that is not go so back in time to admix with another human form, but just admixing between us. You know that all of us were admixed to different extents. One of the most classical cases are African Americans, that are around 70% African and 30% of European ancestry. So we have been asking how much admixture between modern human populations have led to human, or have helped to human adaptation. And for that, we have used our kind of favorite ecological niche, that is Central Africa. We have long-standing collaborations with the Museum for the last 15 years, and we have been studying the genetic history of agriculturalist population from the Central African belt and neighboring rainforest hunter-gatherers. So basically, you know that this transition from hunter-gathering to agriculture has been one of the major transitions to which probably our adaptation to pathogen pressures changed. In Africa, agriculture emerged around 3,000 years ago in this region of Nigeria, Cameroon, and agriculture was transmitted through migrations of people together with languages, Bantu languages, and a few surviving pockets of hunter-gatherers remain, pygmy populations, or rainforest hunter-gatherers in Central Africa, Hatsan Sandawe in Eastern Africa, and Ko Yansan in South Africa. So here the idea was, to which extent, when Bantu-speaking populations that are today 300 million in sub-Saharan Africa, when they move towards sub-Saharan Africa, to which extent they admixed with the local populations they found, hunter-gatherer populations of pygmies, for example, in Central Africa, and if they did so, was this admixture adaptive? So we've been analyzing around 50 populations from different regions of Africa, of both Bantu speakers and local populations of rainforest hunter-gatherers, East African pastoralists, and Ko Yansan. And we have seen that indeed there has been admixture. Here you have the example of Bantu speakers from the West. Each line is an individual, and each individual, as you can see, can have, for example, the first one here, his genome or her genome is 70% red, like 15% gray, and around 5 to 10% red. This shows the different ancestry components in a genome. So you can see that most Bantu populations are kind of red, but they have in their genomes a bit of green, and the green is coming from pygmy populations also of Central Africa, attesting for admixture between these populations. And basically what we have shown is that when Bantu speakers started to migrate all along the Central African belt, they admixed indeed with pygmy populations in the Central Africa and with Eastern African pastoralists in the East. And they acquire, for example, Bantus acquire from pygmy populations some HLA haplotypes, also a form of CV36 that is involved in resistance to malaria. And in the East, Bantus acquire from local populations of East African pastoralists the allele that allow adults to digest milk, so the lactase allele. We have been also addressing another question that in this case it was, we can compare all the genetic architecture of Bantu-speaking populations with populations that result from slave trade in the last 300 years ago, both in America and in South Asia. So by comparing our Bantu populations from Africa to around 5,000 African Americans from North America, we have been able to reconstruct the relative proportions that Bantu populations contributed from the different ports of Africa to modern African Americans. And also we have been studying a population in South Pakistan. As you know, in North America and South America, there's many populations of African ancestry. In Asia, despite the East African slave trade was as important as the Atlantic slave trade, there's much less populations of African ancestry today. But there is a population that is called the Makrani in South Pakistan that is of African ancestry. We have reconstructed using genetics, this case, the genetic history of this population, and we have been able to reconstruct that around 40% of their genomes come from Mozambique. But the question was here, how adaptive or not has been this admixture between African ancestry populations in America or in Asia when they admix it with local populations to search for signatures of post-admixture selection. So we haven't found anything in African Americans in terms of looking at their genomes that are Europeans and African chunks of different ancestry, if there's regions that presents an excess of one of the two ancestries. But actually we have found one in the Makrani from Pakistan that they show around 30 to 40% of their genome of African ancestry, but basically there's a huge excess of African ancestry related to the dark null allele that, as you know, is associated in Africa with almost complete resistance to Vivax malaria. So, but again, not all is about genetics, and we have been also interested in understanding how the lifestyle of a population and the genetics of a population can in turn influence the epigenetic profiles of at the population level. Moreover, there's increasing evidence to suggest that epigenetic changes of human cells can have an important impact on the way these cells respond to infection. So we have been focusing on DNA methylation. And basically what we have shown using this model of DNA methylation by comparing genome-wide DNA methylation profiles in populations of African ancestry that either differ in their recent habitat or in their historical lifestyle, we have shown that recent changes in the habitat of a population, basically transitioning from forest areas to rural areas and to urban areas, will mainly affect processes related to immune response, while historical differences between these populations will mainly affect functions related to development, raising questions about the importance of epigenetic changes in the way we respond to infection. So in terms of ongoing things, now we are exploring a bit more in detail how this Neanderthal ancestry that we have found in our genomes to be associated to the ways we respond to infection, in particular at the level of gene regulatory variants, gene expression, we are looking at how much this is affecting different types of transcriptional regulation or post-transcription regulation, like promoters, enhancers of microRNAs and microRNAs binding sites on the one hand, and on the other hand, we are studying in much more detail how the genome-wide DNA methylation landscape of monocytes first differs between Africans and Europeans, and second, how these differences in DNA methylation profiles will condition the way these individuals and populations respond to viral and bacterial infection. We have shown also quite recently that when you try to quantify the relative contribution of genetics, sex, age, environment in the narrow or broad sense to a phenotype that is very simple in this case to assess, that is transcriptional responses to infection. When I'm saying simple, I'm saying it's not an organismal phenotype like human responses to infection, right? So this should be an easy one. And still, we have seen that in that case, for example, genetics explain just 10% of the variation in transcriptional responses to infection. So a large fraction of immune response variation remains unexplained. So that's why we have now a number of ongoing projects working on different populations that present different lifestyles, are exposed to different environments, including different pathogenic environments. And we're trying to put together different pieces of evidence like genetics, epigenetics, but also microbiota, both nasal swabs and from stools, to try how all this influence immune phenotypes. And with that, I thank the people in my lab, our collaborators, and the funding agencies, and yourself. Thank you.